Well, everyone, hello. Usually I like to come up with some kind of opener for my lectures, but we're on to chapter 6, cell division, mitosis. I have no opener, it's just mitosis. Just cells dividing. And yeah, if you think about it, it gets kind of metaphysical if you think about it, because you are one being, but you're really a collection of 60 trillion, yes, that's trillion with a T, trillion cells that are all molded together to form you, a single organism. Yeah, really, it's just astounding. Um, Variety of tissues that collect together to form organs. And really, what we define as consciousness is really just neurons, brain cells, that have thousands of connections with other neurons and are passing along information, which kind of makes up a biological computer, so to speak. So, yeah, it's amazing. So, cell division. Actually, I did have an opener. I surprise myself sometimes. But, anyways, cell division. Some of these 60 trillion cells that you have, some of them are made when you are born, or made during embryonic development, and then they never change again. They never divide. They never do anything. Some cells divide only when necessary, and other cells divide continuously. So cells divide for a number of reasons. And when I say divide, it's when one cell is turned into two cells. And this happens fairly often for certain types of cells. So let's go ahead now and let's talk about cell division. Now, once again, cell division is the process of cells dividing. They more or less double in population. So cell division is when cell divides, which is exactly in the name, and creates two cells. Now, there are two types of cell division. The one that we'll be talking about is going to be called mitosis. And mitosis is the process where these cells are going to be identical. So if we're referring to mitosis, these cells are going to be identical. I should probably write mitosis here. Now, there's other times where cells divide, and instead of being identical, they are unique. And in that case, that's a process called meiosis. Meiosis is very specific. You won't be learning about that this week, but you will next week. Okay, now why do cells divide? Well, there's a number of reasons. The first is growth. I know that some of you in this class are fairly young, and if you're around the age of 18, 19, just think back a couple of years ago. You were most likely shorter, you were weaker, you didn't have as strong a bone mass. And now, over a period of the last couple of years, you've had spurts in growth, bone density, muscle mass, um, neuronal connections, lots of stuff. And growth occurs when more cells are made. When you are created, you are one single cell. When you are born, you are a small life form. And then as you age, as you mature, your cells are going to start dividing until you reach your genetic potential. So more cells are synthesized. This is an example of mitosis, by the way. So this is why kids can eat so much food. It's because they literally are building themselves from organic molecules. Whenever children eat food, Of course, as long as they don't go absolute ape nuts on the junk food. But when kids eat food, that food is used not only for energy, but it's also used to create new cells. These children grow so fast, especially teenagers like my students. I see what they have for lunch. And like for me, I have the metabolism where if I look at a French fry, I'm already gaining weight. But with them, they're having like French fries dipped in like ranch and then Oreo cookies dipped in, I don't know, dipped in pure sugar. And do you know what? Let's add some deep fried butter to that as well. Reminds me of how much I miss being a teenager. But yeah, more cells are synthesized, which is why children can eat so much food and not become obese. Now, we adults, we still can grow if we eat too many organic molecules, but most of us don't like the direction that we grow in. So, yes, you are capable of growing new cells, usually adipose cells. You can stimulate those to divide if you need to store more energy, and in return, you need to have more synthesis of integument tissue of skin. So yes, you can still grow as adults, but generally, that is not the preferred way to grow if you want an honest assessment of good healthy living. Now the next one is repair, or I should say repair and replacement, but I'll stick with repair. Is damaged or worn out cells are replaced. Some great examples of that. The first is your skin cells, keratinocytes. The skin that you see on your epidermis, your outer layer of skin, is not alive. There's not a living cell on your skin. Instead, the skin cells at the very top of your skin is actually comprised of dead skin cells filled with a strong protein called keratin. Now, what happens is that these dead skin cells, since they're dead and they have no blood supply, they're constantly flaking off, and they need to be replaced by new keratinocytes. So your skin cells are constantly dividing. That's why if you have a wound, in a couple of days you'll find that the wound is starting to heal, and that's because that's how long it takes for the new um, keratinocytes to be converted into mature um, uh, skin cells. Unfortunately, the more these skin cells divide, um, they start to lose quality, to put it simply. And because of that, they're not made with the same, what is say, structural integrity as younger people's skin, which is why older people, their skin becomes to look saggy and worn out and droopy. It's just as a consequence of constant cell division. Those cells start to wear out, in a matter of speaking, and the skin that is made is not nearly as high a quality. All right, and the other is for meiosis, and it would be reproduction. But we won't get into that. Maintenance can go into this too. I forgot to put maintenance on the study guide, but maintenance, pretty sure I spelled that wrong. But maintenance is when cells are replaced. An example of that are the skin cells, the cells that make up the lining of your digestive tract, and they're called enterocytes. What they do is they have a very important job. As food is being absorbed into your small intestines, um, these cells, the enterocytes, they're responsible for absorbing the nutrients that are released during the process of digestion. This process is so important for survival that these cells need to be functioning at optimal strength um, if you want to make sure that your digestive system is functioning as well as it can. So every 7 days, your enterocytes are basically killed off through programmed cell death or apoptosis, and then new ones are synthesized. And why is that? It's because, well, even though nowadays we have plenty of food, in in earlier times when we had prolonged rates of famine and things like that, if you were able to eat food, you wanted to make sure that you can absorb as much of it as possible. And the only way that can happen is if your enterocytes are functioning at maximal ability, which is exactly why um, which is exactly why your enterocytes are replaced every 7 days, because their optimal function is so important. So that's an example of maintenance. So we have something now called a cell cycle, and it is important to note that a cell cycle is not a thing with every type of cell. So the cell cycle, it's important to note that the cycle time is variable for each cell. For some cells, it's really fast, like 24 hours. For other cells, it's measured in months or years. And in other cells, there is no cell cycle because they never ever prepare for mitosis, as we'll talk about. So there's a couple stages of the cell cycle. And the first one is something that we call G1. So G1 lasts quite a while. G1. And G1 is the stage that occurs right after mitosis. So this is a period of normal cell functioning, meaning that the cell has been synthesized. It is now a fully functioning cell, and it's just doing its job, whatever its job is. If it's a enterocyte, it's absorbing nutrients. If it's a keratinocyte, it's being pumped full of keratin protein. If it's an erythrocyte, which is a red blood cell, it's carrying hemoglobin. So G1 is after mitosis. The cell's ready to do its job. It's ready to go. It's also very important to note that non-mitotic cells do not leave G1. So for example, neurons are incapable of undergoing mitosis. There is no mitosis for neurons. Because of that, they will never leave G1. Now, when a cell is going to prepare for cell division, it must pass through a very important checkpoint called the G1S checkpoint. This is a very important checkpoint because this is when various cellular processes determine if the cell is capable of preparing for mitosis. So right now, G1 is the cell that's doing its thing. It's doing its normal functioning. But if a cell is actively mitotic, various checkpoints in the cell, or various check systems, I should say, to not create confusion, various check systems are going to assess if the cell is capable of moving into the stages of cell division. The most important checkpoint is called the G1S checkpoint. If the cell passes the G1S checkpoint, it's going to move to the next stage that we're going to talk about soon. And this stage is called S phase, or synthesis. However, if it doesn't pass the G1S checkpoint, and this is very important, it's moved to almost like a cell purgatory, a side category called G0. And G0 basically means cell is told not to divide. And there's two reasons for this. One is a period of malnutrition. If there are not enough nutrients to build new cells, then obviously the cells cannot be built. So the mitotic cells are told to hold off, to not do anything. This is why malnourished children are smaller in size and stature than their nourished counterparts. The other possibility is that the cell is defective, meaning that the cell is not acting or functioning properly. And if a cell is defective, eventually it's going to undergo apoptosis, meaning it's going to be told to destroy itself, almost like if you watch science fiction shows or movies, self-destruct. So that's basically what it is the cell is told to self-destruct. Assuming the cell passes the G1S checkpoint, it's going to go into the next stage called S, or synthesis. So once a cell passes the G1S checkpoint, it has to divide. If it doesn't divide, if something goes wrong and it can't divide anymore or prepare for division, then the cell will be destroyed. So once it passes the G1S checkpoint, then it is going to have to go through mitosis or be destroyed. So the S phase is DNA is replicated, meaning that you're going to have a completely new set of DNA. And of course, we'll be having an entire lecture or two dedicated to DNA replication. But basically what happens is that once you have one molecule of DNA, then you have two. It's a very complex process. Now the next one is called G2. And in the G2 phase, and this is going to be simple, but cell prepares for mitosis. So things like replicating the necessary organelles, enlarging the cell membrane to prepare to split into two. So the cell makes all the preparatory things to prepare for mitosis. And then finally, mitosis, which actually only lasts a very short time. Mitosis is, excuse me, called the M phase, or mitosis. And this is the series of cell division. Okay, so now we need to talk about some chromosome terminology. So I'm going to show you here. This is what's called a karyotype. A karyotype is a snapshot of chromosomes that everyone has in their body. And chromosomes are only visible during mitosis or meiosis. What is a chromosome? Well, your DNA is actually an extremely long molecule. And DNA exists tightly coiled around these proteins called histone proteins, which is why DNA is compact enough to fit in each cell. Because if you were to take just one strand of DNA, one person's DNA, and uncoil it and stretch it on a table, it'd be about 1 meter long. So obviously that's not the form that we want to store it inside of our cells. So we coil it up, and then when we need to for mitosis to happen, the DNA unravels, and it is going to super condense into something called a chromosome. And I like this picture, even though it's not. Oh gosh, I like the color coded pictures. Okay, well this is decent, I guess. Yeah, no, that isn't good enough. I want it to be numbered. Well, Not the best either. We're going to have to stick with the tried and true here. All right, so chromosomes are pieces of DNA. Think of it like a pizza. If your pizza is DNA, slices of it are going to be chromosomes. So each of these chromosomes, when combined, they form a complete set of DNA. Or I guess another example, if you watch Power Rangers growing up, it's like the individual attack robots that always get their butts kicked. Those are like the chromosomes. But then when they combine into the Megazord, that's like the DNA. If you don't know what I'm talking about, just search for Power Rangers Megazord transformation on YouTube. You'll see what I'm talking about, and you'll go, "Oh, I get it now." So humans, we have 23 chromosomes, but 2 of each. Like think of it as you can have 23 shoes, 23 types of shoes, but you actually have 46 because you have two of each shoe. Now, each of these chromosomes, they differ in the types of genes that they have. We haven't talked about this yet, but a gene is any segment of a chromosome that contains instructions for making one observable thing. For example, there is a part of one of your chromosomes that contains the information for making hair pigments. Maybe it's on chromosome 2. Maybe on chromosome 11, there's the gene for making hemoglobin, which is in your blood. So I guess if you wanted to think of it like this, think of your DNA as a giant recipe book that contains every recipe known to man and woman. But of course, it's hard to find the recipes you want in there. So you're actually going to split that book into 23 smaller books, each one focusing on a different kind of cuisine. Like maybe book 1 will be desserts, maybe book 2 will be Italian cuisine, maybe book 3 will be types of surf and turf, maybe book 4 will be uh, fun with subway combinations. I don't know, but chromosomes are subdivided pieces of DNA. We have 23 individual chromosomes, but we have 46 total. All right, so now that you've seen that, we're going to talk about some chromosome terminology. So what I'm going to do here is I'm going to draw chromosome 1 in purple. So this is what chromosome 1 looks like before. So this is chromosome 1. And this is before S phase. So when I teach mitosis, we basically just go very simple. I usually only use an example of mitosis with chromosome 1 and chromosome 2. But this is what chromosome 1 looks like before the S phase. I generally draw them as shapes because it makes it a little bit easier to teach with shapes than it does with just lines, because all the chromosomes look like lines. So we'll say that chromosome 1 is going to be a column, a rectangle. And you have 2 of chromosome 1s. And you get 1 from each parent. So we'll say mom. And this is dad. What is different about these chromosomes? We'll talk about that later, but each of these chromosomes you have, for example, chromosome 1, is chromosome 1 has the same genes, always has the same genes. So my chromosome 1 might contain genes for hair growth, hair pigment, and hair shape, or follicle shape. But my chromosome 1 has slightly different versions of the gene than your chromosome 1. And to complicate things, these 2 chromosome 1s each contain different genes or different versions of the genes. So, for example, maybe the chromosome 1 from your mom might have the gene for blonde hair pigment. Maybe the chromosome 1 from your dad has the gene for brown hair pigment. The result is that you have the term is dirty blonde hair. But we'll get into that more when we get to genetics. This is just if you're curious about why you have two of the same chromosome. Just one example of it. So before the S phase, you have these two chromosomes that look just like how they did in the karyotype. But after the S phase, something is going to happen here. So this is chromosome 1 after the S phase. After the S phase. So recall that what's going to happen is that, if you recall the S phase, is DNA is going to be copied, right? And what it's going to look like during mitosis is that the chromosomes are also going to be copied. And each copy is going to be attached to the original chromosome through a protein connection called a centromere. So this here is called a centromere. Now, these are identical chromosomes, as in they contain exactly the same genes, exactly the same versions of the gene. And the same thing is going to happen with chromosome 1 from the dad. These are called sister chromatids. Sister chromatids are identical chromosomes that are the result of the S phase of the cell cycle. Oh, by the way, I didn't mention this. Please note this, but interphase is all the stages that do not include mitosis. Sorry about that. So you'll see on the outline it says interphase prepares the cell for mitosis, and it's G1, S, and G2. But anyways, after the S phase of interphase, the chromosomes will be copied. Each copy will be attached to the original, and they'll be connected through a protein called the centromere. It would be like if someone were to clone you and then duct tape your clone to you. That's kind of what's going on here. All right. So now we're going to talk about something called a homologous chromosome. Homologous chromosome. And this is the same chromosome, or I don't like that. Let me change it. Chromosomes that contain the same genes but different variants. Called alleles. And an allele is a different version of a gene. So to give you an example, if we're going to go back to chromosome 1, we'll just say that chromosome 1 from your mom, I think the first one I did was purple. We'll just say that it contains genes for hair shape and hair pigment. And we'll say that chromosome 1, we'll say that one of your chromosome 1s is curly, and I'll just say brown. And let's see that chromosome 1 from your other parent, remember, it's going to have the same exact genes because it still is chromosome 1. But instead of curly, it's going to have wavy. And instead of brown, it's going to be blonde. Now, both of your chromosome 1s, one from mom and one from dad, they both have the same genes, hair shape and hair pigment, but they have different versions of them. We call these homologous chromosomes. because they have the same genes, just different versions of them. Now, on the other hand, there's another term called a non-homologous chromosome. Let me go ahead and erase this. So if a homologous chromosome is chromosomes of the same number that have the same genes, a non-homologous chromosome, there we go. A non-homologous chromosome has two chromosomes, or chromosomes that contain completely different genes. So these are homologous, but let's say chromosome 2, let's say that chromosome 2 contains information for, I'll say, we'll just go ahead and say that it contains information for eye color and I don't know, finger length. So chromosome 1 and chromosome 2 would be considered non-homologous because the only thing that they have in common is that they're chromosomes. But they contain different genes. Sister chromatids are identical, they're clones. And homologous chromosomes are the same genes, or chromosomes that have the same genes, just different variants of them. Non-homologous chromosomes are two completely different chromosomes. All right, so now we're going to go through the stages of mitosis. And we are going to do this model with just two chromosomes. Chromosome 1 is going to be in the shape of a column. So this is what chromosome 1 is going to look like. This is chromosome 1 from your mom's side, chromosome 1 from your dad's side. And we'll go ahead and say that chromosome 2, we're going to go ahead and draw that as a triangle. So chromosome 2, this is chromosome 2 from your mom's side. I know I'm not super good at connecting triangles, but whatever. This is chromosome 2 from your mom's side. Or dad's side, you pick whichever one you want. So what's going to happen here is that the first stage is going to be called prophase. Now let's kind of talk about what's going on with prophase. So prophase is the first stage. Now what happens with prophase is that the nucleus begins to break down. So whenever you see a cell underneath a microscope, the nucleus normally looks like a dark purple spot. You can't see anything inside it. But in prophase, which is the first stage of mitosis, the nucleus begins to break down and the chromosomes become visible. Once again, the nucleus begins to break down and the chromosomes become visible. And I'm not really writing this out because I actually wrote it on the outline. No, I was definitely on my game. You're going to see something called a mitotic spindle form. And the mitotic spindle is a protein connected to two other proteins called a microtubule. And the mitotic spindle has a very important job. It's responsible for both elongating the cell, so mitotic spindle. It's responsible for elongating the cell. And it's also responsible for moving/manipulating chromosomes. Moving, manipulating chromosomes. Chromosomes can't move. Something has to move them. Remember that proteins are the biomolecules in our body that do things. So these proteins are going to act like a fishing rod and fishing wire. It's going to connect onto the chromosomes and move them. It's also going to expand the cell to prepare it to go into two different cells. And we're going to go ahead and we're going to see our sister chromatids appearing for the first time. So we got one. Subsequent ones, I think I'm going to, well, we'll see. I'll just try and keep up the color coding as much as possible, just as long. So we have chromosome 1, chromosome 2 become visible. Now, what's going to happen is that these chromosomes are going to be manipulated by the mitotic spindle, and they're going to be moved to the middle of the cell. And this next phase is called metaphase. And in metaphase, let's go back to the cell here. In metaphase, we have the mitotic spindle is fully formed. And what it's done is it's actually moved the chromosomes into the middle of the cell, a spot called the mitotic metaphase plate. So here's the chromosomes. And keep in mind, these are sister chromatids connected. They are identical, just in case you forgot. Okay. We have our triangles. And if you look here, each of the chromosomes are going to be connected by the microtubule that is connected to the mitotic spindle. Sorry, correction. The mitotic spindle is comprised of a protein called a centrosome, along with, I guess you can call it a wire called a microtubule. You don't need to know the components of a mitotic spindle, but just in case you're curious what these lines are. But you do need to know that its purpose is to elongate the cell, which is what this very outer wire is doing. But it's also to manipulate the chromosomes. And something had to move the sister chromatids to the metaphase plate, and that was the mitotic spindle. So you see that each chromosome is going to be connected to a mitotic, to a microtubule. So it kind of looks like an onion almost. So metaphase, the mitotic spindle has manipulated the chromosomes to the center of the plate. Now the next phase is going to be called anaphase. And anaphase is where you can see the sister chromatids, which will be no more sister chromatids. Instead, they will now just be called chromosomes because they're going to be separated. So you can see that the sister chromatids actually are not going to be sister chromatids anymore. So just give me a moment as I draw these. And we have the triangles. And we have some more triangles. And we have the mitotic spindle connected to each one of the chromosomes. So the mitotic spindle is responsible for pulling apart the sister chromatids and bringing along identical chromosomes now to each side of the cell. So these 4 chromosomes and these 4 chromosomes, they are completely identical. So these 4 and these 4 are identical. And this is all done through the mitotic spindle pulling apart these chromosomes. Now the next phase is going to be called telophase. And in telophase, the cell is going to prepare to make 2 new cells. So if we look in telophase, here we go, big cell, we see the nucleus is starting to reform on each side of the cell. So the 4 identical chromosomes that have been moved from one side to the other, they are going to begin now to reform. So here, and here, and here. So there's going to be a new nuclei that is going to be formed around each of the chromosomes. And then the next phase, which happens really fast, is called cytokinesis. What's going to happen is a protein wire, in this case called actin, is going to wrap around this big cell and it's going to squeeze it into 2. So let's go ahead and draw the end result. So the end result is going to be 2 newly formed cells that are genetically identical. And this is mitosis. So at this point now, you'll no longer be able to see the nucleus. So you're going to have 2 identical cells. Might be helpful if I show you how this actually looks like underneath very powerful microscopes. So let's go to my favorite website, the YouTube. And you can see that, hold on a second. So mitosis cell division. All right, so these are actual images of mitosis. I'm going to try and narrate as it's going on. So we see here, this is prophase. And if you look here, the chromosomes are moving. That's being done by the mitotic spindle. Chromosomes don't just move on their own. And you see that they're being moved into the middle of the cell here. And metaphase is one of those blink and then you'll miss it. Yeah, exactly. Here, let's go back here. So right now, this is metaphase, anaphase. So you see the chromosomes are being pulled apart. And now telophase. You see the nucleus being reformed. Okay. You can see a very similar thing here. This is going to be, okay, I guess they paused it for us. This one is my favorite one because you can really see the entire cell. So this mass here is one cell. You see that the chromosomes are being lined up in the center of the cell. That's going to be metaphase. And there's this stray chromosome here, needs to move to the center. Now, when that happened, we have anaphase. There we go. That's anaphase. And now we're going to have telophase and cytokinesis right there. Now we have 2 cells from 1. Okay, well, everyone, I hope you enjoyed this lecture. This is probably going to be one you'll have to watch more than once. You're probably seeing a trend here. But yes, that is your lecture on mitosis. And next week we'll be having another scintillating lecture on meiosis this time. All right, everyone, as always, reach out with any questions. Over and out.